Welcome to Neet Biodecoded. Here is another question from the chapter Biotechnology. Recombinant DNA technology or rDNA technology was used to produce human insulin, overcoming the challenge of assembling mature insulin. How did Eli Lilly's approach in 1983 successfully achieve this? Let's take a look at the options. The entire insulin gene, including the C peptide, was inserted into the E. coli bacterium where it was naturally processed into mature insulin. Then let's look at option B. Two separate DNA sequences coding for the A and B chains of the insulin were inserted into the plasmids, expressed separately in E. coli, and later combined through disulfide bond formation. Option C. A single DNA sequence encoding both A and B chains was directly synthesized, allowing the E. coli to produce fully functional insulin in a single step. And option D, instead of using bacterial systems, Eli Lilly used mammalian cell cultures to produce insulin naturally, eliminating the need for separate A and B chain assembly. Okay, so by this time, if you know the concept, you would have understood that the correct option is option B, because like human cells, E. coli cannot process a fully functional insulin in a single step. So what happens here is that the two separate DNA sequences which codes for chain A and chain B, they are inserted into the plasmids and they are allowed to express separately and they are later combined using disulfide bond formation. So this is how the Eli Lilly's approach to producing mature insulin. Option A is not right because the entire insulin gene, including the C peptide, cannot be processed as an E. coli. That's what I explained. And a single step process cannot be achieved also. This is also wrong. And option D, where they're using mammalian cell culture is wrong because they opted for E. coli because that is more beneficial compared to using mammalian cell culture because the maintenance of mammalian cell culture is far more expensive than a E. coli system. So by looking at the option B is more accurate. So this is the answer. So keep preparing well for your examinations. I'll see you with another question.